DNA gets all the glory, but right now, at this exact moment, your DNA isn't actually doing the heavy lifting to keep you alive. That job belongs to a workforce of over 100,000 different molecular machines, physically building, repairing, and operating your cells. Welcome to the world of proteomics, the scientific discipline dedicated to mapping this invisible, incredibly complex cellular workforce. Here is the paradox at the heart of biology. Almost every cell in your body contains the exact same DNA sequence. The instruction manual is identical. Yet the brain cell on the left looks and behaves entirely differently from the muscle fiber on the right. Having the blueprint to a house doesn't tell you how the plumbing works, what the building materials are, or who is actually swinging the hammers. To understand the house, you have to look at the workers executing the plans. Those workers make up what we call the proteome. Unlike your genome, which is locked in place, your proteome is highly dynamic. It shifts second by second, reacting immediately to a sudden spike in temperature, a viral infection, or even the stress of missing a meal. The process starts when DNA is transcribed into RNA, which is then translated into long, linear chains of amino acids. But a straight chain of amino acids is useless. To actually perform physical work, these linear strings must collapse into their lowest energy three-dimensional shapes. Their lifespan is heavily regulated through a continuous cycle of creation and destruction. When these proteins are damaged or simply no longer needed, specialized disposal complexes called proteasomes tear them apart and recycle the pieces. So, while your genome remains practically identical your entire life, your proteome is a real-time, physical reflection of exactly what is happening inside your body right now. The challenge for scientists is a logistical nightmare. How do you capture, isolate, and measure millions of shifting, invisible machines that are all operating at the same time? The first step is extraction. In this diagram of a technique called 2D gel electrophoresis, a chaotic mixture of cellular proteins is placed in a gel and pulled horizontally across the x-axis by an electrical current. Because different proteins carry different electrical charges, they organize themselves into distinct columns. Then we add the second dimension. A new current pulls those same proteins downward through a polymer matrix. This time, they are separated by their physical size and mass, leaving us with a cleanly separated map of individual protein spots. With the proteins isolated, we bring in a tool called a mass spectrometer. Lasers shatter these individual spots into smaller, manageable fragments known as peptides. The mass spectrometer then uses precise magnetic fields to measure the exact mass of these fragments. The result is a highly specific numerical readout, a peptide fingerprint unique to that exact machine. Through these physical extraction techniques, a biological machine is successfully converted into pure, measurable numerical data. Raw mass data by itself tells us very little. Interpreting it requires serious computational power and an enormous catalog of historical data to reference. This brings us to the massive public sequence databases, global repositories managed by institutions like the National Library of Medicine that hold every genetic and protein blueprint ever sequenced. We feed our new peptide fingerprint into bioinformatics software using an algorithm called BLAST. As shown here, the query sequence stays anchored on the left, while it rapidly searches against millions of known sequences in the database until a statistical match locks into place. Once that match is confirmed, the algorithms assign our mysterious protein to a known functional family and accurately predict its final 3D structure. In this pipeline, bioinformatics acts as the digital link between raw mass and biological function, reconstructing fragments of data back into a map of a living, working cellular machine. On a human scale, mapping the proteome offers a direct look at active physiology. While DNA outlines the genetic risks you inherited, the proteome reveals the specific biochemical processes occurring in your body right now. This is the basis of forward proteomics. By directly comparing the mapped proteome of a healthy cell against a diseased cell, like the network on the right, we can easily spot the cluster of nodes glowing red. Those red nodes represent abnormal protein expression. Isolating these specific red flag anomalies allows researchers to discover biomarkers. These are the earliest physical warning signs of conditions like cancer or neurodegeneration, detectable long before symptoms appear. Identifying these specific faulty proteins allows pharmacologists to design targeted therapies. By focusing on the broken molecular machines themselves, researchers can develop treatments tailored to the specific protein imbalances driving a patient's disease. Genomic data predicts a patient's potential risks, but proteomic analysis captures the active state of their disease. For many complex conditions, this real-time data provides the clarity needed to select the most effective treatment path. But targeting a single protein is just the beginning. Every single one is a node in a sprawling, interconnected network of thousands of simultaneous reactions. Biologists call this vast interactive web the molecular sociology of the cell. It's a space where mindless, individual proteins coordinate their actions to form a living, breathing system. The genome provides the blueprint, but the proteome provides the function. It's the true architecture of life. If you want to see more of how this invisible architecture shapes our world, hit the subscribe button, leave a like, and check out our next video to keep exploring.